Hello and good afternoon. Welcome to the first IMIAS webinar. I'm Peter Korsten, Department Chief of Rheumatology and Clinical Immunology in Germany and Associate Board Member of IMIAS. We are very happy that you are connecting. We are still waiting for you to come in and let other people enter the webinar. And I'm really happy to have this first inaugural webinar with a very special guest, Iago Pinal Fernandez, who's a staff clinician at the NIH and a assistant professor at Johns Hopkins in Baltimore. And Iago, you're most famous for interfering in myositis, but that's truly not half of the story, and I'm really looking forward to your talk today. And please keep your cameras off. You'll have the chance to ask questions through the chat after the talk, and it will be open for discussion, and I hope you will engage with us. And great to have a increasing number of participants still dialing in. And Iago, I'm excited to have you here. The floor is yours. We're looking forward to your talk.
Okay, let me share the screen. So thank you so much for the invitation. So it's really exciting to be able to talk to you about some of the things that we have been doing at the NIH and with our collaborators all over the world. Today, I'm going to talk about some of the things that we have worked in the very recent times. So some of the things that I'm going to show you are things that we saw just a couple of months ago. So I'll really welcome all of your feedback and any ideas that you may have on how to move this work forward. So first, I'm going to start talking about one of the things that made me more curious about myositis. So when I started seeing patients with myositis, it very quickly became obvious that these patients are not all the same, even within the same syndromes. So we have patients with dermatomyositis, the antisynthetase syndrome, immune-mediated necrotizing myositis, and overlap myositis. In this talk, I'm not going to talk about IBM, which I think is in a different category. But when you have seen enough patients with dermatomyositis, you see patterns that are very clear. So, for example, one of the most striking ones, anti-MI2, anti-NXP2, anti-TIP1 gamma patients, they usually don't have lung involvement, but patients with anti-ND5, they have very striking lung involvement, sometimes fatal. So when I came here to the NIH, we spent a few years doing epidemiologic analysis, looking at what are the patterns of each one of these autoantibodies. But a few years later, we started asking the big questions. So why are we seeing these patterns? Why the antibodies are different among themselves when they're in the same syndrome? So today I'm going to talk to you about some of the evidence that we gathered about what is the role of the antibodies in the disease. So one of the conceptual barriers to understanding the function of antibodies in myositis was that antibodies, it was thought that if they had to do anything to disease pathogenesis, it had to be done in the outside of the cell. Because we know well that proteins don't get across the membrane of the cell very easily. And we have verified that. When you put antibodies into cells in culture, you see very little effects. But today I'm going to show you some of the mechanisms that we think are maybe relevant to this phenomenon of the antibodies getting inside of the cells, and why we think this may be more important than what it was previously thought. So to do this, I'm going to focus in two of these conditions. The first one is going to be anti-MI2 dermatomyositis, and the second one is going to be anti-PMSCL overlap syndrome. And the reason why I'm going to focus into these two is because the data is much more clear, and there is a good reason for this. And the reason is related to what we do, which is transcriptomics. And these particular two antibodies are very related to transcriptomic effects. We have seen effects also in many other different antibodies, anti-MD5, anti-synthetase syndrome. But those, if you have questions, I'll be happy to answer them, but I'll be focusing on these two first. So the first one is anti-MI2 dermatomyositis. So anti-MI2 antibodies were the first ones to be discovered in dermatomyositis. And the antigen of these antibodies is this multi-protein complex that is located in the nuclei of the cell. In particular, MI2 proteins are CHD3 and CHD4, these ones in red in the middle. And this multi-protein complex, its function is to bind to the nucleosome and repress the expression of genes. So this is the machinery of the cell that keeps the expression of genes that are not necessary in a certain cell type off. So for a long time, we have been doing this technique called RNA sequencing. And what we do is we get pieces of muscle, we smash them, we extract the RNA, and we prepare a library. And what we get with this technique is a snapshot of what the muscle is producing at a given moment in time. Just for you to get an idea of the magnitude of the data that we get with this technique, we get about 60,000 individual measurements for each piece of muscle. And this is only looking at genes, pseudogenes, and first category items, not looking at transcripts where we see much more. And what I'm going to show you includes 814 muscle biopsies, including healthy and disease controls. But many of the things that I'm going to talk about, we realized when we had the first batch of sequencing out of the more than 8 years ago. So our approach has been to look into the things that are specific for each one of these groups. So instead of looking at the differences between each one of the antibodies and controls, which they have thousands of differences, some of them are very unspecific that you can see when you have genetic myopathies, or you can see when you have infectious myopathies or other types of myopathies where antibodies don't have anything to do with the pathogenesis. Our strategy was to look at the differences between, in this case, anti-MI2 and the rest of the groups, normal, IBM, IMNM, anti-synthetase, and EM, and look at the things that are shared across all of these comparisons. So when we did this a long time ago, we saw that there were about 135 genes that were unique for anti-MI2 antibodies. Here you can see how these genes look like. Here in the x-axis, you have NT is normal muscle. Anti-MI2 is the anti-MI2 dermatomyositis. And here you have an XP2, TIP1 gamma, MDA5. They're also dermatomyositis, but they are not expressing these genes. So we're wondering why this specific gene set is being overexpressed in this particular antibody. And the first clue that we had was from animal models. So there were several animal models where they had knocked out the MI2 protein in the muscle and in the heart. And what they saw is that some of the genes, about 50% of the genes that we had detected, came up when you knock out the MI2 protein in the mouse. The second clue was a little bit more interesting. So we were finding genes that were related to the function of the autoantigen. So anti-MI2 antibodies and the genes that we were seeing were related to the function of MI2. So the second thing that we tried to address is, okay, if the antibodies are doing anything inside of the cell, they need to be inside of the cell, right? So we looked directly at where the antibodies were in the muscle biopsies of our patients. And I can tell you that when we saw this, it was really, really exciting because we weren't expecting to see it as clear as we saw it. So this is a muscle biopsy of a patient with anti-MI2 dermatomyositis. And here each one of these circles is a muscle fiber cut cross-sectionally. And here on blue, you have a marker for nuclei. So you can see that wherever there is a nuclei in this biopsy, there is green. And green is a direct immune fluorescence for immune globulin. So this is where the immune globulin is accumulating in the muscle of patients with anti-MI2 dermatomyositis, in the nuclei of the cell. So here you have one nuclei, another nuclei, another nuclei, and this was pretty broad. So we were seeing in some of the biopsies more than 95% of the nuclei had detectable accumulation of immune globulin. So the next thing that we have done, this is more recently, we have looked if the same happens in the skin of these patients. So this is a skin biopsy of a patient with anti-MI2 dermatomyositis. And you can see in blue is a counterstaining for the nuclei, but in green is the immune globulin. And you can see that the nuclei of these patients are accumulating immune globulin as well. And then we wondered ourselves, so how can we confirm that the antibodies are the ones that are causing the effects that we are seeing in the human muscle? So to do that, we developed a technique where we culture normal muscle cells in the lab. We put the antibodies purified, so we get the serum of the patient, we purify the antibodies, and we put them in the supernatant of the cells. And then we use electricity to make pores in the membrane and get the antibodies inside. It's a pretty artificial model, but it is very simplistic, so we can analyze the exclusive effects of the antibodies in the cells. And when we did this in anti-MI2 dermatomyositis, we saw something that to me was pretty striking. So these are the results of the experiment. Each column is the purified immune globulin coming from a different individual. So here you have 4 controls. Here you have 5 patients with anti-MI2 dermatomyositis, 5 patients with PMSEL, and you have NXP2, T1 gamma, MDA5, the rest of the dermatomyositis. So when we put the antibodies, the purified antibodies of patients with anti-MI2 inside of normal muscle cells, we saw that all the genes that I had previously shown you, so these are just the top genes that we had seen in the muscle biopsies, all of them start coming up. All of them get upregulated. So we have this situation where we have antibodies against something that is in the nuclei of the cell. We're seeing antibodies in the nuclei of the cell. And the effects that we're seeing in the muscle biopsies, we are able to recapitulate them by getting the antibodies inside of the cell. And then, and this is the part that is a little bit more recent, we did this Shinium spatial transcriptomics. So this is a technique to look for RNA in the muscle biopsies. Unlike previous generations of spatial transcriptomics, the main advantage of this technique is that the spatial resolution is amazing. So how the technique works is that you get a slide with muscle biopsies, and then you put probes, and each one of these probes, and this is why the spatial resolution and the specificity is so nice, they have 2 different sequences. And these 2 different sequences, they need to be very close together, so you can do this rolling cycle amplification, and you get a lot of RNA for that particular type of RNA that you're measuring. And then you use fluorescent probes, and you look where each one of these fluorescent probes is located. One downside of this technique is that you cannot measure the whole transcriptome. You have to be very, very selective on what you measure. And we were extremely selective. We use actually the smallest version that they sell, and we measure 380 genes that are part of an immune oncology panel. And on top of that, we put 100 genes. And these 100 genes were genes that were interesting for the muscle, like the ones that I'm showing you here, which are the different types of myosin that identify the different muscle fiber types. And the other types of genes that we added were the genes that were specific for anti-MI2, there were genes that were specific for anti-PMSCL, and some other genes that were interesting for our purposes. So this is just this slide showing you the different myosin heavy chain types associated with the different muscle fiber types, is to show you what is the spatial resolution of this technique. So here you have myosin heavy chain 7 in green, you have myosin heavy chain 2 in red, and myosin heavy chain 1 in white. And you can see how the borders of the muscle fibers are really well defined. Just to zooming in one of these regions, you can see that each one of the dots is a different RNA that was detected. And you can see that the space in between the cells is empty. So you can even define the things that are in between the muscle fibers. So now I'm going to show you the same way that I saw it the first time that we did this technique. And it happened in December, so it's not a long time that we have had this data. How we saw the things that we were interested in. So now I'm going to switch to the Shinium Explorer, which is a tool that allows you to visualize this kind of data. And what I'm showing you here is the specific gene set for anti-MI2 dermatomyositis. So these are the specific anti-MI2 genes that we had identified in the muscle biopsies that we know that are induced by the antibodies in the cells in culture. And this is looking at a muscle biopsy of a patient with anti-MI2 dermatomyositis and looking at these specific genes. So you can see that there are different colors, and this is because each one of these, they all appear in the same type of muscle fibers. Now I'm going to zoom out a little bit, and you're going to see what is the distribution in the muscle of these transcripts. And you can see this is a fascicle. And one of the characteristics of anti-MI2 dermatomyositis is that the involvement is usually prefascicular, like other forms of dermatomyositis. And you can see how these specific transcripts are located in the prefascicular region. Now I'm going to zoom in again in this cluster of fibers. Well, the first thing that was very, very striking to us was how selective this transcriptomic signature was. You see that there are these 4 fibers that have lots of these transcripts, but the fibers that are around, they don't have a lot of the transcripts. And you see these 2 yellow fibers. These 2 yellow fibers, yellow is a counter-staining for RNA, so this is staining for 18S. And you can see that these 2 are not expressing lots of these transcripts. These are regenerating muscle fibers. And these are probably new fibers that are being made that still didn't get the effects of the antibodies. So now I'm going to tell you some of the things that we detected with this technique. The first one, we didn't know if the antibodies were toxic to the cells. And one of the things that all the muscle fibers need to make is myosin heavy chain 1, 2, and 7, which are the, as I told you earlier, the markers of the different fiber types. And as you can see, these 4 fibers in the middle, they're not making myosin heavy chain 1, 2, or 7. So this transcriptomic signature is really, really toxic to these fibers. We have other fibers that are still expressing the transcripts, and they are still making the myosin heavy chain. So that is one of the reasons why we think that the transcriptomic signature precedes the development of muscle damage. Another transcript that is interesting to look at this is titin, which is another gene that is usually expressed at very high levels in all the muscle fibers, and you can see that these ones have less of it. Another thing that was puzzling to us is that when we internalized the antibodies, we didn't see interferon. And interferon, as we all know, is one of the key characteristics of dermatomyositis. So now I'm going to show you type 1 interferon inducible gene, one of the most indicative ones, which is ISG15. And if you take a look at ISG15, the fibers that are around these 4 ones that have the transcriptomic signature, they have lots of interferon. They are being affected by the interferon at very high levels. And the ones that are regenerating, they're one of the ones that are being affected by the interferon the most. But the ones that are affected by the effects of the antibodies, they are not doing that. And that was interesting to us. If we look at and I think we have a clue of why this may be happening. So if we look at the receptor of interferon, you can see that the expression of the receptor of interferon is pretty clearly visible in the cells that are around these 4 fibers here, but they are not very clear in them. So one possibility is that these fibers cannot sense interferon because the receptor of interferon is turned down. Another thing that we had observed, so one of the specific genes that we had detected was interleukin 11. And interleukin 11 is not one of the most striking ones, but what we didn't realize at the time is that interleukin 11 is downstream of TGF-beta. And TGF-beta is extremely toxic to the muscle. It causes muscle atrophy. And here you can see the expression of TGF-beta. You can see how it is restricted to these muscle fibers here. And you can also see that it is also present in the regenerative muscle fibers that you have here, the yellow ones that I was telling you before. And this is probably the reason why we didn't identify the TGF-beta when we did the bulk RNA sequencing, because it is physiologically overexpressed in regenerative muscle fibers. So for example, patients with immune-mediated necrotizing myositis that have lots of regeneration, they're going to have lots of TGF-beta because they have lots of regenerating fibers. But what we are seeing here is something different. Is the direct effect probably of the antibodies. And we have seen the same when we internalize the antibodies into the cells in culture. Okay, so this was the part that we were expecting to see. We were expecting to understand a little bit better why we weren't seeing ISG15 in the muscle fibers. We were expecting to see if these antibodies were toxic or not to the muscle fibers, and understand a little bit better what were the inflammatory pathways that were implicated in the disease. But then we saw things that we weren't expecting. So you can see here a thing that is very yellow in the middle of these 4 fibers. And this thing, as I was telling you, yellow indicates that there is lots of RNA. So this cell here, if I'm gonna start showing you the markers of the plasma cells, for example, this is SYNDECAN1, this is C38, and this is the pseudogene of the immune globulin, which until I started analyzing this data, I didn't know that it was a good marker for plasma cells, but it is one of the best ones that I have detected, that we have in the panel. So this is an antibody secreting cell, most likely a plasma cell. You have here another one. This was not the interesting thing, finding plasma cells close to the muscle fibers that were having the transcriptomic effects. What was interesting is that we had included in the panel the immune globulin RNAs. So I'm going to show you the IgG kappa sequence. And now I'm going to show you the IgG one, the heavy chain. And now I'm going to zoom in this particular cell. And I'm going to remove the CD38, the SYNDECAN, and the pseudogene of the immune globulin. And this is what struck us as interesting. So this is the plasma cell. And we all know that plasma cells make antibodies. But what we were not expecting is to see the RNA of the immune globulin not being inside of the plasma cell and being outside of the plasma cell and inside of the muscle and in the space in between the muscle fibers. This we weren't expecting. And there are very few mechanisms that can explain what we are seeing, which is sequence specificity. So we are seeing some RNAs that are getting out of a cell and some RNAs like SYNDECAN1 and SYNDECAN1 and CD38 that are not getting out of the cell. One of the few mechanisms, the only one that I know of, is extracellular vesicles, exosomes. And it is known that plasma cells can make exosomes. It is known that plasma cells can make lipid vesicles that are externalized and they are loaded with immune globulin protein. But what nobody had studied probably was if they were loaded with immune globulin RNA. So I think one of the possibilities is that these plasma cells are just making these exosomes loaded with immune globulin protein, and that immune globulin protein is the reason why we're seeing antibodies inside of the muscle fibers. Another more exciting possibility is that these exosomes may be loaded with the instructions to make the immune globulin. This RNA is being transported through vesicles to the other cells, and the immune globulin protein may be made by the other cells. There is still a lot of work that we need to do to clarify this, but this was one of the things that was very striking to us. And here I'm going to remove SYNDECAN1 and CD38. And you can see that there is a plasma cell here, which is an IgG kappa. There is another plasma cell here, which is another IgG kappa. And moving here, this fiber here is highly affected by the transcriptomic effects of the antibodies. And you see another plasma cell here. And in this case, it's not an IgG. You don't see any blue here. This is an IgA kappa. So there are plasma cells from different isotypes. Now I'm going to change the color of the IgA to red. So you're going to see that this plasma cell is making IgA and the kappa chain. And these RNAs are also getting inside of the surrounding muscle cells. So this was very, very interesting. But then we wanted to go a little bit farther. So we have these plasma cells that are internalizing the immune globulin RNA into the other cells, and we are seeing transcriptomic effects that are caused by the internalization of the antibodies. And now we wondered, how do the anti-MI2 antibodies disrupt the function of the NURT complex? So this was, we figured this out in a very interesting way. And again, we weren't expecting to figure this out this way. So we were looking for other antibodies. in patients with different types of myositis. We had actually included the anti-MI2 patients as positive controls. And we did this technique called FIB-seq. And the FIB-seq stands for phage immune precipitation sequencing. And what this technique, how this technique works is that you have phages that have the sequence, and you made phages for all the different sequences of the whole proteome, different peptides, one at a time. And these phages, they have the sequence of the peptide that you are encoding for in the inside, and they have the peptide in the outside. So then what you do is you use the antibodies of your patients to fish these phages, and then you sequence the DNA sequence that they have in the inside, and you can see what antibodies they have based on the sequences that you detect. And so it's basically a way to detect new antibodies. When we did this technique, we identified that anti-MI2 patients, they had antibodies against AIR, which is the protein that is dysfunctional in patients with APESET, which is autoimmune disease that causes immune dysregulation and endocrinopathy. And this is the validation. After we did the FIB-seq, we validated these findings using both ELISA and a new technique that we are using, which is called LIPS, luciferase immune precipitation assay, which has orders of magnitude more dynamic range than in ELISA, allowing us, for example, this is 5 standard deviations above the control group, so the signal-to-noise ratio is much, much nicer. Here you have control, anti-MI2, and the different forms of myositis, and you can see that all the MI2 patients are also positive for antibodies against AIR. Here you have also IBM, lupus, systemic sclerosis, vasculitis, and Sjögren, and you can see how clean the signal is. So patients with anti-MI2 antibodies, they all have antibodies against AIR. And then we wanted to answer the question, it is that they have two different antibodies against MI2 and antibodies against AIR, or it is that they have antibodies that recognize both at the same time. So to answer this question, what we did was we affinity purified antibodies against MI2, and then we tested them in an ELISA for MI2 and ELISA for AIR. And what we found is that the anti-MI2 antibodies, the ones that only recognize MI2, they also recognize AIR. So it's not that these patients have two different antibodies, it's that they have one antibody that recognizes different proteins. And then we went back to the FIB-seq. The main application of FIB-seq is to discover new antibodies, but it has a second application, is that since you are doing tiles of peptides, you can identify what is the epitope that is being recognized by these antibodies. When we went back to the FIB-seq and looked at what was the region of error that was identified in most of the patients, we saw that it was this region here, 271 to 360. And when we went and looked for the regions that were recognized by the antibodies predominantly for MI2 beta and MI2 alpha, it was this region here that was the most common one. When we looked at these sequences, what we found is that they corresponded to the same functional domain, the PhD domain. So this PhD domain, it is very interesting because it is where the mutations happen in AIR causing APESET. And it is one of the key regions of MI2, and I'll talk about this a little bit later. So here, you remember at the beginning, I told you that MI2 binds to the NURT complex, right? And the MI2 antibodies bind to the NURT complex, recognize the NURT complex, and that this NURT complex needs to bind to the nucleosome. The way this NURT complex binds to the nucleosome is through MI2 proteins through the PhD domain. So I think what we are seeing here is an antibody that binds to the PhD domain, doesn't allow the whole complex to bind to the nucleosome, and then it cannot repress the expression of genes. And we are seeing genes that shouldn't be expressed in the muscle. One of the most striking ones, for example, is insulin. So the muscle of these patients is making insulin RNA, and this causes disease. But what is interesting is that I told you about AIR, I told you about the MI2, but there are many other proteins in our body that contains a PhD domain. So we wanted to go a little bit farther and see, okay, there are about 80 proteins that contain a PhD domain, including MI2 alpha, beta, AIR. What about the rest of them? So for that, we first selected one that we just selected at random, SP140L, just to see if any of them, if we selected one at random, would bind to these antibodies. And there is another one that is very interesting, which is TIF1 gamma. So TIF1 gamma, which is the antigen of a different autoantibody in dermatomyositis, also has PhD domains. So let's start with SP140L. When we did a LIPS to detect antibodies against SP140L, and we actually developed the technique to detect only the PhD domain of this protein. And when we did the test to detect the PhD domain of SP140L, most of the anti-MI2 patients detected this peptide. So patients with anti-MI2 antibodies also recognize the PhD domain of SP140L. And what about TIF1 gamma? So to do that, we did several LIPS assays. The first one is using the full-length protein. Here you have controls, and the controls, they don't bind to TIF1 gamma. They don't have antibodies that bind to TIF1 gamma. You have patients with anti-MI2 dermatomyositis that they bind to TIF1 gamma with intermediate binding affinity. And then you have the patients with anti-TIF1 gamma antibodies that actually bind to TIF1 gamma with a lot of affinity. So this has been a historical problem in antibody detection. Patients with anti-MI2 and TIF1 gamma, they cross-reacted, and we didn't understand why. And probably the reason is because we are trying to make antibody assays that try to differentiate between these two antibodies when both of them bind to TIF1 gamma. So if we try to make a cutoff to differentiate between these two, we can draw a line here, and we have to choose if we want to have a bad test for TIF1 gamma, if we want to have a bad test for MI2, or a bad test for both. But we cannot have a good test for both using the full-length protein of TIF1 gamma. What we did was, okay, so if the anti-MI2 antibodies are binding the PhD domain of TIF1 gamma, if we remove the PhD domain, we should see things more clear. And we did precisely that. So we removed the PhD domain. So this is the TIF1 gamma without the PhD domain. And without the PhD domain, the anti-MI2 antibodies stop binding to it. But the TIF1 gamma antibodies still bind to it. So this may be a way to improve some of the antibody tests by understanding what are the epitopes that are relevant to each antibody. And now I'm going to switch gears and go to anti-PMSCL in the last few minutes of the presentation. And this story is a little bit simpler. And it is a little bit simpler because of the function of the autoantigen in anti-PMSCL. The antigen that the antibodies bind to is this nuclear RNA exosome complex, which is a multi-protein complex that is located in the nucleoli of the cell. The main antigens are exos 9 and exos 10, which is the enzymatic part of the complex. And its function is to chop long non-coding RNAs, divergent transcripts, antisense RNAs that our cell doesn't need anymore. And this is a key part of the cell. It's kind of the recycling bin of our nuclei. And as I told you earlier, these are located in the nucleoli of the cell. So we use the same techniques that I explained in detail for MI2. First, we did bulk RNA sequencing, and we identified what are the specific genes that characterize this specific antibody. And we identified more than 200 genes. As you can see here, the names of these genes are different than the names of the genes that we were seeing in anti-MI2 dermatomyositis. They all start with the NSG because they don't have a name. And the reason why they don't have a name is because many long non-coding RNAs don't have a known function and nobody spent time putting a name on them. So many of these are long non-coding RNAs. The second thing that we did was look at the muscle biopsies of our patients and look where the antibody is deposited. So here, as I was showing you earlier, this is a counter-staining for the nuclei. It's just telling us where the nuclei are. And in green, we're seeing where the antibody is deposited. And you can see that there are some dots inside of the nuclei. I'm going to zoom in one of these dots. So this dot is actually the nucleoli of the cell. So the antibodies are accumulating in the nucleoli of the cell, different to MI2 that the antibody was depositing in the whole nuclei. And we saw this in the muscle but recently we had also the chance to do a skin biopsy of a patient with anti-PMSCL antibodies, and we were able to see it also in the nucleoli of the keratinocytes. The keratinocytes, the nucleoli degrades as they grow, so we were seeing some fragmentation, but you can see that there is antibody inside of the nuclei and a distribution that reminds the nucleoli. And then we did the same that we did with the MI2. We got serum from our patients, we purified the antibodies, and we were able to recapitulate the effects that we were seeing in the muscle biopsies of the patients. So here you have all the long-non-coding RNAs that we had detected. Here you have some of them that end in DT, which are divergent transcripts, and some that end up in AS, that those are antisense transcripts. And you see more DTs, more antisense here. So this is basically the substrate of the antigen that these antibodies are binding to. And it is accumulating. It is indicating that the antigen is not working well. And you can see how specific it is. These are 5 different patients with PMSCL after 3 days after the electroporation. And you can see the control, the MI2, the XP2, the rest of them are not making these transcripts. And then we did the spatial transcriptomics. And we designed this spatial transcriptomics also to see for the anti-PMSCL specific genes. And you can see in a muscle biopsy, in this case, the muscle fiber is cut longitudinally. And you can see how this muscle fiber has many areas where these transcripts are accumulating. If I zoom in this area here, you can see that actually these anti-PMSCL specific genes are accumulating within the nuclei of the cell. And this makes sense. The action, the effect of this antigen is happening in the nucleoli of the cell. So the RNAs are accumulating in the nuclei of the cell. They cannot get out until the cell is very damaged. The second thing that we did was look for the things that all the muscle fibers need to make it they are healthy, like myosin heavy chain 1, 2, and 7. And you can see that this fiber here that has lots of these transcripts is not making any of this. Is making much less than the surrounding fibers. So this muscle fiber is really, really affected. And then we looked at the plasma cells, similar to what we did in anti-MI2. So this is a region of the muscle biopsy that is heavily affected by the antibodies. You see all of these transcripts that are being overexpressed. And in the surroundings of these muscle fibers, there are plasma cells. The IgG kappa here is in green, the IgG is in red. This is an IgG kappa cell. This one is actually an IgM kappa. I'm not showing you the IgM. So there are many plasma cells, and if you see the plasma cells longitudinally, there are multiple plasma cells around the areas that are affected. So I think we are getting close to kind of a general framework for how myositis works, for how myositis, what is the pathogenesis of myositis. As I was telling you, there are these plasma cells that probably are mediating the internalization of the antibodies, or at least are part of the story. It may not be the full story. These are probably internalizing the antibodies through exosomes, which is a known function of the plasma cells. What was interesting is that we identified that the exosomes were probably loaded with RNA. Then these antibodies that are being internalized bind to the autoantigen, and they block the function of the autoantigen, causing cell toxicity and inflammation. But then the question is, okay, I told you about MI2 and PMSCL. What about the other forms of myositis? Is this exclusive for these 2 types, or we can see it in others? So what I'm showing you now is a muscle biopsy of a patient with anti-TIM1 gamma dermatomyositis. And one of the main characteristics of these patients is that there is lots of type 1 interferon in the perivascular region. And you can see here, there is a flap of fascia that was detached. It was originally close to this. And when we look at the expression of plasma cell markers, you see how all of the plasma cells are living here. So perhaps what we were seeing as perivascular involvement, perivascular overexpression of ISG15 was just translating that the plasma cells that are making the antibodies are in that particular region. So the cells that are close by are the ones that are being affected the most. Now I'm going to show you a different biopsy. This is a patient with anti-MDA5 dermatomyositis, and there is lots of ISG15, MX1 overexpression, lots of type 1 interferon signature. And you can see where the plasma cells are located. And you can see that as I was showing you before in the MI2, there are genes that don't get out, and then the immune globulin genes, they get out and they get into the surrounding cells. All of these are plasma cells. This is a massive plasma cell infiltrate, and they are all around the areas where there is ISD15 and MX1 the most. And finally, what about the other forms of myositis? For example, this is a patient with anti-HMGCR, necrotizing myositis, and myosin heavy chain 3 and 8 are good markers for muscle regeneration, which muscle necrosis is one of the main characteristics of these patients. And you can see here a cell that is yellow, is making lots of RNA, and this is a plasma cell that is externalizing the immune globulin, the IgG heavy chain and the kappa light chain into this area that is affected. So probably from the data that we have, it suggests that this mechanism also affects the rest of myositis. But what about other forms of autoimmunity? So we have just started to approach this, but when we did direct immune fluorescence of patients with anti-U1RNP that had mixed connective tissue disease, you can see how all the nuclei are accumulating immune globulin. And this was actually proposed in the 80s. It was one of the first autoimmune diseases where it was proposed that internalized antibodies were actually pathogenic, but I don't think that they had the tools to see it very clearly. So this is URNP, which is mixed connective tissue disease, a condition in between lupus, scleroderma, and myositis. This is another one, anti-KU. And you can see how all the nuclei of the cell are accumulating immune globulin. This is a centralized nuclei that has a bunch of immune globulin. And this is an overlap between lupus and myositis. And what about pure forms of other autoimmune diseases? So this is anti-SCL70, one of the classical antibodies in systemic sclerosis. And this is a skin biopsy of a patient with anti-SCL70 systemic sclerosis. And you can see how all the nuclei of the keratinocytes are accumulating immune globulin. So I think, as I have told you, I think this mechanism is very, very relevant to myositis, but perhaps is a little bit broader than that, and it affects other autoimmune diseases and is relevant to other autoimmune diseases. So now I want to thank all the people that made this possible. I'm not including all the people that was implicated in all the different experiments, only the main ones. But our group at the NIH, Dr. Catherine Pack, Dr. Casal Dominguez, Sandra Munoz, that were key to doing all this spatial transcriptomics and transcriptomic analysis that I told you. Our sequencing core that was key to all of the things that I was showing you. Our collaborators at the Clinic Hospital and the Valdebrón Hospital in Barcelona, Dr. Milicenda, Dr. Selva O'Callaghan, the group at the Mayo Clinic, the group in Berlin. And now for some of the things that I was telling you, we have been able to validate them outside in collaboration with our colleagues in Canada, with our colleagues in France. So this is a growing group of people that are helping work this up and provide information about what is actually driving the disease. And now, if you have any questions, I'll be happy to answer them.
Thank you very much for this truly exciting talk, Iago. You took us through a very interesting journey of your experimental data in myositis and probably relevant to other autoimmune diseases, as you mentioned. So just for the participants in the audience, you can also raise your hands and ask questions directly. We're about 40 participants, so feel free to ask any questions. The first one is from the chat. Can you read it, Iago? Do you have the chat open?
Let me.
It's about
okay,
HLDR, myofiber expression and correlation between HLA-DR expression.
So the question is, in the neuropathology work we have done looking at HLA-DR myofiber expression, there were a correlation between myofiber type and HLA-DR expression. This may be investigated and explained by your techniques. So yes, I didn't tell you, but in the PMSCL, so we have seen that HLA type 2 is associated with interferon gamma pathways. So one of the conditions where the interferon gamma pathway is relevant is anti-PMSCL, and we have seen both interferon gamma signature and HLA type 2 in this particular type of muscle biopsies. We have also seen it in IBM, and with the techniques that we are using, spatial transcriptomics, we are able to detect it in other types of myositis where it's very hard to detect through conventional histology.
Then the next one from your honor, are there specific immune cells that co-localize with plasma cells?
Yes, that is a great question. So close to all of the plasma cells, there are T cells, CD4 T cells, that are probably the ones that were implicated in maturing them from B cells to plasma cells. One thing I didn't tell you is that this externalization of immune globulin RNA only affects plasma cells. It doesn't affect B cells. So the B cells, the IgM doesn't get out of the cell. There are also other things that are interesting. For example, I didn't show you, but in anti-MI2 dermatomyositis, there are macrophages that are also subject to this transcriptomic profile, and they are making interferon beta. We can detect interferon beta itself in some of these macrophages. So it's not only the muscle is where it is easy to see these effects, but we are also seeing it in, for example, PMSCL in fibroblasts and endothelial cells. In the MI2, we can see it in macrophages, and in the lab, we can internalize them and see the same effects in many different cell types.
Okay, next one is a comment in the question, where can we find a paper that summarizes your great talk?
So I can share my screen again, and you can. So you have this accession number. We have it in preprint, so you can read about some of the things that I told you related to spatial transcriptomics. It is publicly available, which is amazing for something that we saw for the first time at the end of December. So that we were able to put it publicly so quickly. I really like the preprint strategy.
If people are looking for your papers on PubMed, they will find a lot of the work that you have been publishing and reviews on the topic and so forth. But we can, after the webinar, we can also put some links to your, if you identify some key papers that you want people to know, or maybe you want to promote your work a little bit more, we can add them to the
I can put the
show
link
notes,
for
the
link to the preprint that contains most of this in the chat. Yes.
Perfect. And then there's another question. How does the transcriptional data correlate with H&E staining? Do you see normal fibers in H&E with abnormal transcriptomics? And how can it be maybe detected to detect subclinical disease?
Yes, so one of the beautiful things about the spatial transcriptomics is that they have this counter staining that is very similar to the H&E. So if I share the screen and go back to the spatial transcriptomics, you can see that these fibers, if I remove all the color, they look pretty healthy. By H&E, I would say that they are normal. There are some of them that don't look normal. This would be what we would consider pre-necrotic fibers. But the ones that I was focusing on by H&E would be classified as normal. And I didn't talk about this before, but in previous studies, we have correlated the intensity of this transcriptomic signature in MI2 with the histology of the muscle biopsies, and it correlates pretty nicely. So the patients that have the highest expression of these genes, they are also the ones that have more inflammation and more muscle necrosis. So it matches really, really nicely.
Then there's another one on your purified antibodies. Are they total IgG, or were they purified by a specific antigen that possibly on selected IgG? Were the patients absent of other myositis-associated antibodies, or did they have one specific antibody?
Yes, so we didn't do affinity purification for the internalization. One of the challenges is that for the internalization, you need quite a bit of immune globulin. So it is challenging to get the affinity purification. What I was showing you was just the IgG purified with protein G, and we internalize all of them at the same time. There are some things that we are investigating, for example, interactions between different antibodies, for example, anti-Gro52 with other specificities that seem to be interesting. But the strategy that we chose was to choose different patients that have the same specific antibody and see if we see the same thing or not. But you are right. The clearest approach would be to do affinity purification, but getting affinity purified immune globulin at the levels that we need for internalization has proven to be challenging.
And then maybe I can just drop in a question myself. So you nicely show that internalization of autoantibodies causing disruption of functional proteins is a causative mechanism in those diseases. And probably, do you think we are doing good enough with our treatments, which not necessarily work in specific compartments? For example, rituximab, extracellular activity. Do we need more therapies like daratumumab, which enters the compartments, or CAR T cell therapy earlier, a deeper B cell depletion in those diseases? As you showed, autoantibodies are quite causing disease in those myositis subtypes that you showed.
Yeah, so one of the things that I didn't discuss, but I think is fascinating, is how well what the story that I was telling you matches with what we see from the therapeutic world. So anti-B cell therapies, they should work actually, because the plasma cells do not live for a long time. Most of the plasma cells, they tend to die in a couple of months. And that is probably why Rituxan is not very quick to do the effect, because it doesn't target the plasma cells, it targets the B cells that are the precursors. Yes. And then you need to wait until the plasma cells die. One question that I still have is, what is the role of long-lived plasma cells? And they're having strategies with CAR Ts where the CD19 CAR T was not effective, and then they use a BCMA CAR T, and that was effective. So it may be that some of these plasma cells living for long periods of time may be the cause why some of the CAR Ts don't work. But I think all the effective treatments that we have, CAR Ts, you were talking about daratumumab, teclistamab, all of them work probably because these cells are the ones that are driving the disease. I think now we have a unique opportunity to not only be able to treat the disease, which I think teclistamab CD38 may be very effective to do that, but also to go a little bit earlier, target the B cells and reset the immune repertoire. The question is, how deep are we resetting the immune repertoire? If we are getting rid of all of these clones, or some of them are still hovering around and we're going to see flares after a few years.
And you also mentioned myositis testing. I think there's a problem in routine clinical work that we are only most antibodies, we don't have quantitative, well-established tests to monitor disease activity. I mean, it would make sense in some diseases, or to some extent, to have antibody levels declining with therapeutic response. Just like in HMG, CR antibodies, it does correlate quite well in my experience. But in most patients, we only use myositis immunoblot tests, and we have only semi-quantitative information on how well the expression of antibodies is in a particular patient.
And there is another problem that we are using binding assays. We're using assays that tell us if the antibodies bind the protein or they don't bind the protein, but they don't tell us if the antibodies cause a functional effect. So, for example, one of the clearest examples coming from infectious diseases, where there is a form of autoimmune disease that targets interferon gamma. And what they do to diagnose the patients is they first do a binding assay to see if the patient has antibodies, but then they do a functional assay to see if these antibodies block the function of the binding of interferon gamma to the receptor. So I think we have a unique opportunity here to move away from the binding only strategy and start doing functional assays. I think some of the problems that we are having is because some of the patients, they have the antibodies, but they don't do any harm to the cell. So you may have anti-MI2 antibodies, I show you in the FIP-6, that are binding to some of the regions outside of the PhD domain. They are not causing disease. So they are just, we would consider them false positives, but they are actually binding the protein.
So then we will move on with the questions from the chat. The next one is, I have several patients with either huge hypergammaglobulinemia, either poly or monoclonal, who develop myopathy-like, and for who we identified Ig deposit on myofibers. Any idea on this non-specific Ig-mediated myofiber dysfunction?
Yeah, so this is a very interesting group of patients, so patients that have multiple myeloma and develop autoimmunity. May it be that the antibodies that the multiple myeloma is making are the ones driving the autoimmune disease? That I don't know. We don't have big cohorts of these patients, but they are super, super interesting. Another evidence about the internalization of the antibodies is if you see biopsies from patients with multiple myeloma and amyloid, some of the accumulations are inside cells. So that was another line of evidence that the antibodies could actually enter the cells.
And next one is, do you data on CD38 overexpression in the fascia of TIF1 gamma reconcile with MRI findings in dermatomyositis that show predominance on inflammatory signals in the fascia as well as muscle pain reported specifically in this subset?
Yeah, I think they do. I showed you the spatial transcriptomics that all the plasma cells were in the fascia, and as Ocean was saying, when we do the MRIs of patients with TIF1 gamma and other dermatomyositis, we tend to see involvement in the fascia and pain and elevation of aldolase. So I think this may also be explained in the MRI, not only the histologic perspective.
So then there were a couple of hints to your link that you sent, but I think there's a correct link to your preprint. Yes, for me it works. The last one that Corinna Preusse posted is working. So I don't see any more questions or comments. If people have particular questions, I think they can approach you directly by email. Everybody knows how to reach you. And thank you very much for this truly exciting talk, very interesting data. And I think it was a great premiere and very honored to have you here on our first series of iMyos webinars. And I hope you stay tuned for the next episode. Thank you very much. Have a good morning, day, afternoon, wherever you are. Bye. See you next time.
Bye.